Welcome everyone, my name is Nicole Farfán, and in this space I will briefly explain the research carried out with Molina, Ibeth, Romero and Jair Téllez from Colombia, which is titled In Silico Characterization of Cysteine Synthase Enzyme in Pseudomonas aeruginosa as Possible Therapeutic Target in this Pathogen. To get into context, Pseudomonas aeruginosa is a strictly aerobic gram-negative bacillus that is ubiquitous in nature. This pathogen has been related to healthcare-associated infections, representing between 10 and 15% of deaths, mainly affecting immunosuppressed patients. Its treatment has been affected by the different resistance mechanisms that this bacterium uses to block the action of carbapenems, uses as a first-line antibiotics against this type of infection. For this reason, the need has been created to search for new target of action focused on the development of the new therapies, which is why in this study cysteine synthase was proposed and studied at the in silico level for that purpose. Cysteine synthase is an enzyme of importance in the regulation of cysteine biosynthesis, mainly in the second part of the novel biosynthetic pathway, which is present in bacteria, parasites, and plants. Cysteine synthase acts with some cofactors such as pyridoxal phosphate and sulfides to obtain cysteine, which is essential in the structure, folding, and assembly of proteins, and is also a basic block for the tire synthesis in parasites and bacteria, important in the protection against oxidative stress. The search and the identification of the sequence of the gene that encoded for cysteine synthase in Pseudomonas aeruginosa was carried out through the free database on the GenBank website at NCBI, and the motifs present in the sequence were obtained with ScanProcyte. These sequences were compared with orthologous cysteine synthase enzymes present in other important microorganisms by means of a multiple alignment using the ClustalW tool of the bioedit package, and a phylogenetic tree was constructed with the MegaX software using the amino acid sequence to analyze the evolutionary relationships that exist among their orthologous. Subsequently, the 3D structure was modeled with iTaser, and the general and local quality of the structure of this model was evaluated through the analysis of the graphs obtained in the PROSA web server and in a project. The results showed us that the Pseudomonas aeruginosa there are 3 gene sequences that code for cysteine synthase: cis-A, cis-B, and cis without characterization. Only the typical pyridoxal phosphate binding motifs were found in isoform A and B, and the third isoforms did not yield any hits, so this could be a pseudogen. The multiple alignment and phylogeny shown in the first image showed that cis-A and cis-B were more related to the orthologous present in Escherichia coli and Salmonella typhimurium, and its relationships with the closest orthologous in the human, which is cystathionine beta synthase, has less than 13% similarity, which is important when proposing it as a target of action. The 3D model of the Pseudomonas aeruginosa cysteine synthase enzyme obtained in iTaser is in the row A of the second image, and the TMNC scores indicated that the global topography of the structure is correct. The model validation graph is in the row B on the second image, in which we can observe that all the C-scores for the 3 isoforms are within the typical range of native proteins with similar size already described crystallographically by nuclear magnetic resonance or X-ray. Finally, in the row C of the second image, we can observe the validation that was carried out to the secondary structure by means of a Ramachandran graph, in which it can be observed that the more than 85% of the amino acid residues are found within the allowed and accepted regions, and that are observed that in yellow and red. So it was concluded that the structure is stoichiometrically stable. Thanks to the study, it was concluded that bioinformatic and comparative analysis of cysteine synthase from Pseudomonas aeruginosa suggests that the novel pathway may be present as the main biosynthetic pathway of cysteine in these microorganisms, and cysteine synthase from Pseudomonas aeruginosa can be good drug target because it's not present in humans, and the closest orthologous in humans, the cystathionine beta synthase enzyme, is very low in similarity. Thanks for your time and attention. Bye.